To understand how it is LTP takes place, you have to think about what happens at a synapse where the axon terminal releases glutamate. So let's picture an axon terminal, postsynaptic dendrite or dendritic spine. Here's the axon terminal. It's full of vesicles. In each vesicle is about 7,000 molecules of glutamate, or some number close to that. Right? And then in the postsynaptic surface, here, let's call it a dendrite in this case, you've got glutamate receptors. So the receptors for glutamate are present here in the postsynaptic membrane. The axon terminal releases glutamate. So that's what we have right here. Here we have the postsynaptic membrane, and in it are glutamate receptors. Now glutamate is an interesting neurotransmitter because, sure. It has ionotropic receptors and it has G-protein coupled receptors, just like just about every neurotransmitter. There are some that only have GPCRs, but there's no neurotransmitter that just has ionotropics. Okay, so this is showing you the ionotropic receptors for glutamate, and that's where glutamate is unusual because it doesn't have one. It has 3 types. Now, the ones that we've mainly talked about when we talk about the fact that glutamate gets released, so release here, produces an EPSP here, and you can record it reliably every time. Well, that's glutamate having bound to AMPA and kainate receptors. AMPA and kainate. AMPA is an acronym. It's no big deal. No one remembers the whole thing except the P stands for propionic acid, and no one cares about that. It is named for the fact that these receptors bind this compound called AMPA really well. Similarly, kainate receptors are named for the fact that these receptors bind another drug called kainic acid really well. Okay, so these are the guys, the AMPA and the kainate receptors, produce these rapid EPSPs. You see them every time, reliably enough. What about these guys? So NMDA is also an acronym, stands for N-methyl D-aspartic acids. NMDA is a drug that binds really well to glutamate receptors, but only some of them. And those that NMDA binds really well to are called, therefore, NMDA receptors. All 3 of these are glutamate receptors. Some bind AMPA better, some bind kainic acid better, some bind NMDA better. And the NMDA receptors are the big deal because these guys are strange. They do not work unless 2 things happen. So we think of NMDA receptors as being the detector of a coincidence. And the coincidence is, number 1, the presence of glutamate. They have to bind glutamate. So that's number 1, they have to bind glutamate. But here's number 2, glutamate remains bound to these receptors for about 20 milliseconds. That's pretty standard for a neurotransmitter. We'll bind to a receptor and stay bound for 20 milliseconds. So it's true. So glutamate has to be bound to an NMDA receptor for its channel to open up. But number 2 is the postsynaptic neuron has to generate an action potential. So within those 20 milliseconds, while the NMDA receptor has bound glutamate, the postsynaptic neuron has to generate an action potential. Well, if we think about that, when does an axon, this is an axon, ugly axon, but it's an ugly axon. Anyway, when does an axon release glutamate? When an action potential invades the axon terminal. That's what causes neurotransmitter release. And then over here we have an NMDA receptor, and it doesn't work until it binds glutamate. But at the same time, within the same 20 milliseconds, the postsynaptic cell has to generate its own action potential. So the NMDA receptor detects the coincidence of a presynaptic action potential leading to the release of glutamate and a postsynaptic action potential leading to activation of the NMDA receptor. All right, so that's the coincidence. Coinky dinky. Coincidence. Presynaptic action potential followed 20 milliseconds later, within 20 milliseconds. It doesn't have to be exactly 20 milliseconds, but within that period of 20 milliseconds, while the glutamate has still bound to the NMDA receptor, the postsynaptic cell also generates an action potential, and the NMDA receptor detects that coincidence and it opens. It does. It will open in response to release of glutamate by the presynaptic neuron and its binding to the NMDA receptor. There's glutamate, followed within 20 milliseconds by the postsynaptic cell generating its own action potential. That is the coincidence that NMDA receptors detect. As a result of that coincidence, the NMDA receptor channel will now open, and it will allow ions to pass through it. And you ought to be asking yourself, big deal. Well, the answer in that is, which ions?